Hello students, in the last lecture we discussed about basic terminology of inheritance including gene, DNA, codon, heterozygous, homozygous, dominant, recessive, genotype and phenotype. So today we are going to discuss about monohybrid cross, sex determination in human, codominance, and pedigree. These are our basic topics that we are going to discuss. First of all, I will discuss about monohybrid cross. You know, monohybrid cross is the cross between two individuals in which we only select one character, in which we consider only weight. Like in this example, we are considering here two plants. One plant is tall while another one is dwarf. Dwarf means short. So this is parental generation, right? So if you see here, this condition is homozygous dominant. The phenotype for this plant is tall, right? So it means this is tall, right? So it means we are doing cross between tall and dwarf, right? So we are doing a cross between tall and dwarf. So first of all, gamete should be formed by this plant. So if you see here, the genotype of tall plant is T and T. Both genes are dominant. So if it is going to form gamete, so what will happen? By the process of meiosis, it will form gamete. So one T will go to one gamete, right? Gametes are actually 6, and this T will go to another gamete. So it means two gametes will form and both are same because both having same allele, right? Both having T and T, both are capital T. It means both are dominant. Same for dwarf this plant. Same for this plant. If you see here, here we have T, right, and small t. Both alleles are recessive. Now, if it is making gametes, right, by the process of meiosis, gametes will form. Remember, as I told you already, that when the organism is going to form gametes, process of meiosis will happen and diploid number of chromosome will convert into haploid. It means here double setup. It means double number of chromosome. Here for example, here we have two alleles, so these two alleles will separate from each other. It means 2n number of chromosome will convert into n number of chromosome, right? So same is like same. By that concept, these two alleles will separate from each other because these two alleles are present on two different chromosomes, right? So one chromosome will go to one gamete and one will go to another. So these two alleles separate from each other. So we get two gametes. But if you see here, with respect to this allele, both gametes are same. And here also these two gametes are same. So we can take, for example, this can give me this T gamete, right? If this T gamete fuse with this small t gamete, means this dominant fuse with this recessive. the process of fusion is called fertilization. It means if this gamete come and fertilize this ova, so what will happen? It will give me heterozygous plant, and this plant is still tall, right? Why it is tall? Because this T is dominant over this recessive one. According to Mendelian genetic, it is complete dominance here. So this T, this capital T is dominant over recessive one. So the phenotype of this offspring is what? Tall. This offspring is tall. And this is the generation, the first generation that we get from parental generation, that is called F1 generation. So this is the offspring of F1 generation. Now, we can also do cross between, for example, if we want to see that if we do a cross between the offspring of F1 generation. So the offspring, the genotype of F1 generation is what? T and small t. So here you will also take a T and small t, right? So these two are two individual, means these two are two, like this is one plant and this is another plant. Both are the offspring of, uh, both are the offspring that we get in F1 generation from parental generation. So what kind, how many kind of gamete it can form? It can form two kind of gamete, right? And that T will go to another gamete, right? Same will, this will also give us T and T gamete and T gamete. So let's suppose one is male, right? We consider it male, and another we consider it female, right? If it is in plant, so it means it is, for male it is pollen grain, and for female it is ova. So this sign, you know, this sign is for female, right? This sign is for female. So in this side we take what? Female gametes, and in this side we take male gametes, right? T and T, T and T. So let's suppose this gamete fuse with this T. So what we will get? We will get a plant that having heterozygous, homozygous dominant condition, and the phenotype of the plant is tall. And let's suppose this T, this gamete fuse with this gamete. So we will get T and small t. This condition in heterozygous condition, and this plant is tall. Okay. Now let's suppose if this T, this gamete fuse with this. So what we will get? We will get T and small t, again heterozygous condition, and this plant is tall. If this plant, this gamete fuse with this gamete, so what we will get? Small t and small t. So both are like the condition is homozygous, and both allele are recessive. So it means this plant is dwarf. So if we see here, what is the phenotypic ratio? This, remember, this is called Punnett square. This table is called Punnett square. So from that this Punnett square, we know that what is the phenotypic ratio. So phenotypic ratio is we have 3 tall, right? So we have 3 tall, while this one we have dwarf. So 1 we have dwarf and 3 we have tall. So the phenotypic ratio we have 3:1, right? While the genotypic ratio, give the genotypic ratio for it. So genotypic ratio for it is what? So homozygous dominant tall is how much? Both are capital T, 1, right? And heterozygous, how much we have? 2. So T and small t, right? And how many homozygous recessive we have? 2. No, sorry, 1, right? So you write it here 1 and small t and small t, right? So remember this is 1, okay? So what is the genotypic ratio? 1 to 1 ratio. 1 to 1 ratio. It is genotypic ratio for this cross. I think it is clear to all. Now Now, this is another example for monohybrid cross. In this, we already get F1 generation from parental generation. So in this case, if you see here, this is this is the seed color. Seed color is the phenotype. And for the trait that we select, it is for seed color. So we have two type of seeds. One is green color and another is yellow. So remember, yellow is dominant over green. So we are going to represent yellow by capital Y, while the green we will represent it by small y, right? So you know the green color is recessive, while capital Y, the yellow color, it is dominant. So let's suppose if a condition is like this. So it means the condition is heterozygous because here two different alleles are present, right? While the phenotype of this plant will be, or this seed color will be what? It will be yellow, right? Why? Because the big, like the capital, this is yellow. The capital Y express and that suppress the this recessive allele, right? So here we have two heterozygous individual, two heterozygous seed, right? And both color is yellow phenotype. And when we do a cross, so what will happen? So I told you how many gametes will form from it. One capital Y and small y, right? For it. And for this capital Y and small y, we write gametes on one side. Now do the cross. If this cross with this, so we will get y. This cross, right? This cross with this, right? So we will get capital Y and small y, right? So, and if this cross with this, so what we will get? Capital Y and small y. Capital Y is supposed to be first, right? And then small y should be here. And if this cross with this, so what we will get? Both are small y and small y, both are recessive. That's why we get it here, both small y and small y. And this is green, while this is capital Y, this is yellow, capital Y and small y. This is yellow. Why? Because it is dominant over small y. And here this is also capital Y and small y. Capital Y is dominant over small y, so it is yellow. So again, if you see here, the phenotypic ratio is 3:1. 3:1 means we have yellow seed, how many? 3, while the green we have 1. While the genotypic ratio, so you can see here, genotypic ratio, homozygous dominant, homozygous heterozygous condition, while homozygous recessive. So how many dominant we have homozygous? 1. Heterozygous, 2, right? And recessive, how many we have? 1. So 1 to 1 ratio again for this class we have. I think it is clear, right? Now let's move toward another. This is six determination in human. I told you already in my last lecture that there is 46 number of chromosome, that is a 23 pair, right? So for everyone, these chromosome are homologous. Chromosome 1, chromosome 1, chromosome 2, chromosome 2. It means this person get from father, this one from mother, this from father, this from mother, father, mother, right? Like this up to 22. Same here also, one from father, one from mother. But you know that this 22 pair, that is 44. These are called autosomal chromosome. They having no concern with 6. While the last pair, this show the sex of an organism, right? Like how in this example, like, and female, the last pair, 20th pair, this is same, both. That's why we represent it both by XX, right? While in male, one is short, another is long. So one is X, just like the female, right? And another is Y. So we represent it by X and Y. So if a person get X from father, sorry, yeah, it's okay if it get from father, right? But no, like if a person get X from father and X from mother, so what will happen? That again, that person will be, that offspring will be female. While if a person get Y from father and X from mother, so that person will be male. So remember, female only give X chromosome containing gamete, while two types of sperm will form in male, one X and one Y. So if X fertilize the OVA, there will be female. If Y sperm fertilize the OVA, there will be male, right? So now you can check it in this cross. So let us do about, you can see here, this is monohybrid cross. So in female, we will represent one side, for one side, this is for female, okay? So this side is for, this side, this is for female, right? And this side for male, right? It means sperm are here, right? So you can see here. So and female, I told you there will be XX, right? So if XX, so definitely how many type of gametes will form? Only one type of gamete will form. X, X chromosome containing OA and X chromosome containing OA. So we wrote it here already, XX. Same for male, how many type? Two types of gamete. Some sperm may contain X, some sperm will contain Y. Now let's suppose this X fertilize this sperm. So what will happen? XX will form, right? If this X fertilize this OA, so what will happen? X and Y will form. If this X fertilize means it fused with this sperm, so X will form, right? And this, if it fused with this, so what will form? X and Y. So it means that there is chances of, you can see this is, so let's see, this is female, this is female, while this offspring is male and this offspring is male. So the chances is how many? Like you see, 2, right? So 2 means 50-50 probability, 50-50 chances is there. If a mother is giving birth to a baby, so there is 50-50 chances that it may be male or that that maybe she maybe a female, right? Male or female. So the chances is 50-50. I think it is clear now. Okay. Now let's move toward another. Okay. So this is same as I did to you, as I did now, right? Like XX, so X. And for male, why they selected only one here in this class? Because don't be confused here. If we select it here, because it is only same, if I put it here X, again it will give me 50-50 result. So just to make it easy, for that reason in some book they mention only one, like a gamete is forming same kind of gamete, so that's why they just mention only that X. Okay. And here is different types, so then we must mention that too, because these two are different, right? So if you do it, so 1-to-1 ratio means what? 50% and 50%. So same result, right? Is it clear? So you just be remember that like parental, P for parental phenotype, right? So it is female, parental genotype is XX, and when gamete forms, so this is X, right? And after that, what will happen? Fertilization will happen. For example, if this X fertilize this one, so female will form. If this Y fertilize this X, so male will form. Hope it is clear. Now, what is codominance? Codominance is a condition in which two allele express in heterozygous condition. Two allele of specific gene express in heterozygous condition. A single gene has more than one dominant allele, and both of that allele express equally. Right? I will give you now example. If you see here in this example, you can see phenotype for this hen is white, black, and speckled. Genotype is WW, BB, and BW, right? So WW, it is a recessive, right? This is white color. While B for black, it should be this black color, right? Now, when heterozygous condition came with us like BW, right? B and W. So according to Mendelian genetics, it's supposed to be like this, that this B should suppress this W, right? And this should be black. But no, what happened here? It's another phenomena happen that's called codominance. In codominance, both of these two allele of specific gene express equally. And we get a hen that having, you can see, you can look at here to this chicken, like here is black color and on that there is white. So it means this black for this B and white for this. We can differentiate clearly. So this is codominance, both express equally. Same as in case of blood group. You can see here, especially AB blood group. You know, like for example, there is three allele per blood group. IA, IB, and IO. These three allele for the blood group we have, right? Now let's suppose if a person get blood group allele A from mother, okay, and he get allele IO from father. So the person blood group should be A. Why? Because A is dominant over IO. If a person get IA from mother and IA from father, so this condition is homozygous dominant. Both are A and A. So a person again blood group should be A. Remember, this A represent us, in this case, it represents as the antigen A on the surface of red blood cell. For example, this is red blood cell on the surface of it. This is antigen A, right? Now let's suppose a person get B from father and IO from mother. So what should be the blood group? Again, this condition is heterozygous. So B is dominant over IO. So the person blood group should be B, right? Let's suppose if a person get IB from mother and IB from father. So remember, this is B, okay? So this blood group should be again B, right? Why? Because this is homozygous condition, homozygous dominant, B and B. So the person blood group should be B on the surface of red blood cell. For example, this is red blood cell, there will be another type of antigen, not like A, right? Now come to our codominance. Let's suppose if a person having IA, he get from father, and IB he get from mother. So what should be the phenotype of this? Which antigen should be on the surface of red blood cell? So remember, it's the example of codominance. So both antigen will, both allele will express, and both antigen, this is antigen A and this is antigen B. Both express it here, means A also express and B also express, and we can see it here. This is for A and this is for B. So this condition is called what? Codominant, in which both allele express equally in heterozygous condition, right? Now remember, some people are getting confused with incomplete dominance, right? So incomplete dominance, they, like in that condition, also both allele express equally. But the problem is that an incomplete dominance intermediate form, you cannot differentiate between that. This expression is of which allele. Let's suppose I cross red flower, right? For red, I will represent it R, right? Red flower with white, and I will represent small r for white, right? when I do it, so what happen when I do a cross? So that I get F1 generation, R and R, right? Now, according to Mendelian genetic, is supposed to be this R should suppress this one and it's supposed to be read. But in incomplete dominance, what happened? It give me another intermediate between these two intermediate phenotype. It give me that is pink. So this is incomplete dominance. Why it is incomplete dominance? Because I don't know that this R, which is the expression of this in red, but it give me a middle intermediate between red and white. Right here, I know here is a clear difference that for A this is antigen, for B this is antigen. Here for B this is black, and for W it is the. But here I it's just give me intermediate. It give me the intermediate between these white and red. So this is the example of incomplete dominance of it is clear to all. Okay, if we are writing mostly the codominance and capital letter. Okay, now this is a very nice example of codominance. You can see it here, mother and father, and when the son having both, like both strip, right here, both strip it here. So this is a very nice example for codominance, right? Both express, mother genes also express, mother allele also, father also express, and offspring, the baby having both strip, right? Now come to a TESTCROSS, right? So before going to TESTCROSS, let me tell you about what is actually TESTCROSS, and I will give you example of that. First, learn example. Let's suppose if I have a red color in my hand, right? Let's suppose if I have a red color flower in my hand, and I tell you, you know already that red color is dominant, but I tell you that it is heterozygous or homozygous. So then you will unable to give me the answer of that. So in that case, what you will do, you will cross that flower with homozygous recessive. Homozygous recessive is, for example, white flower. So you will cross with that. If you get all the all the offspring red, like all offspring, you do the cross and then you get all the offspring red color, 100% red. So let's see. So what does it mean? For example, I have red color flower, and for that, if their genotype I don't know, but I'm just taking an cross. So I have to cross my red color with recessive one, and recessive is RR, right? And when I do a cross, so what I get all the offspring like R small r, R small r, all the R small r, right? Remember, all these are individual. R small r, R small r, R, sorry, R small r small. So four offspring came with me, and it means that all the offspring are red, right? Because this R is dominant over small r, right? It's complete dominance, remember. So if all the offspring, if the flower that was in my hand, and when I cross it with a recessive, when I cross it with the recessive, and all the offspring came with me red color. So it means the flower that was in my hand, it was homozygous. That condition was homozygous dominant. But if I cross it with homozygous recessive and I get 50% red and 50% white, so it means the flower that was in my hand, the genotype of that flower is heterozygous dominant. How it will be like, for example, you can see heterozygous dominant cross with recessive flower white. So when you do a cross, so R, it means this R came with this and this R with this. You can also make a checkerboard peanut square. You can also make it by that, and this R with this now, and right, and this R with this now. So it mean that I get 50% red and I get 50% white. So from this, if I get 50% and 50% red, so from that I will conclude that the flower that was in my hand, whose genotype is that is heterozygous dominant, right? So this is test cross. Remember, in test cross we always do cross with recessive, homozygous recessive. Okay. Now I will give you another example for the test cross. Okay, now you can see here, this is the example for the test cross. Let's suppose this dog having black color, right? Black color is dominant over, they mentioned chocolate color, but I will mention brown color. So the black is dominant over brown color. So if I do a cross between these two, and I get all the upspring black, so from that I will conclude that this dog having a homozygous dominant genotype. If I do the cross between these two and I get 50% black and 50% brown color, so from this I will conclude that the the this black dog genotype is dominant, right? And I did a class to you already about it before. Understood? Hope it is clear to all. Okay. Now let's move towards pedigree. You know what is pedigree? Its pedigree is actually family tree that explains your genetic history. Pedigree are used to find out the probability of a child having disorder in a particular family. To begin to interpret a pedigree, determine if a disease or a condition is autosomal or X-linked or dominant or recessive. To determine to interpret a pedigree, first we should know that the disease gene, the disease that happened because like the disease happened because of gene, so that gene is present in autosomal chromosome or that is present on six chromosome. Six chromosome we have two type X link. If a disease is like if the disease gene is present on X chromosome, so we call that X-linked disease. If it is present on Y chromosome, we call it Y-linked disease, right? But mostly it is X-linked because X chromosome is long and they contain more gene as compared to Y. Y is short. Hope it is clear, right? Now, and then we will also know that if the disease happened because dominant or because of recessive. It means, like for example, normal gene is dominant. If that normal gene converted into recessive, so that disease is recessive. If the normal gene is recessive and that converted to dominant, and after mutation it caused disease, so that is what dominant disease, right? Now let's see, we will learn from the example. First of all, you should know about the pedigree samples. These are the samples. You know, this is if this sample, this square without shade show us normal male. This circle show us normal female. If it is shaded, it means the male is affected. If this is female, right, shaded female, so it means the affected female. While if it is half shaded, right, it is carrier male. You know, carrier is a person who having the defective gene, they that having the ability to transfer that defective gene, but usually carrier having no symptom of that disease. It means he is normal, but he has still the ability to pass that defective gene to their offspring after mating, right? And this one, you know, this one is parent mating, like parent, like male mate with female, right? And this is sibling. This show you brother and sister, right? Is it clear? Now let's suppose This is example. Is the affected allele dominant or recessive? From this, what do you know? It's dominant or recessive? Yeah, let us see. It's dominant or recessive. Yes, the affected allele is recessive. It is what? Recessive. Why? If you see, all the recessive are having disease. So in this condition, the normal gene was dominant. But when mutation came and that normal gene convert into recessive, because of that disease happen, and that is recessive, right? Now let's see another example. In this case, what is it? The affected allele is dominant. Why it is dominant? Because if you see, all the dominant having a disease. So it means before disease, the normal gene was recessive. No problem was there to the person. But when it, because of mutation, when it converted dominant, and when that express, so that affect a person, right? All right. So this is dominant. Now, uh, the disease, genetic disease may be autosomal, or it may be a six leg disease. Autosomal means it's present on autosomal chromosome, as I discussed you already before. Except X and Y, all other chromosome are autosomal chromosome that control our body character, right? If and the disease may be six link. Six link, I told you already, it may be X linked or it may be Y. The gene that is present on six chromosome, and problem came in that gene and that caused disease. So that is called six link disease. If it is on X chromosome, this is called X linked disease. If it is on Y, it is called Y linked disease, right? Here they give you example that X is equal to normal allele. But when mutation came and this X, so we represent it by disease recessive allele. But it is not must. Its representation depend on you, how you want to represent it. No problem, right? You can see here, they did a cross between normal father, X and Y. This is normal. With with carrier mother. Why carrier mother? Because mother having this normal X, but she having defective gene. She having no symptom, but she having the ability to transfer to their offspring. When they did a cross, so the offspring that came with us, normal daughter, normal son, carrier daughter, and hemophilic son. Hemophilia, it's hemophilia is a condition in which a person is unable to do blood, like he or she cannot, having no ability to uh, clot the blood, right? A minor injury, too much bleeding can happen from a person, right? So that condition is called what? Hemophilia. So it means that hemophilia is a recessive. Why it is a recessive? Because the dominant gene convert into recessive one, and we represent it here by R. So this person get what? One R. So this become, there is no homologous, only Y. So this R express in this male, and he become a hemophilic patient. But here this female, this carrier daughter, she gets this XR for defective gene from mother, but she get X from father, that is normal. So that normal is dominant over this recessive. That's why they didn't express in this and her daughter, right? Now this is example of, I told you already, six link disease can include hemophilia and colorblind. Colorblind, a person unable to differentiate between the colors, especially red and green color. Now let's see how we will know. Here, a person having a disease, I will tell you, like, for example, I will take the example of XR. So this person, this is male, right? So this male having a disease because it is shaded. I told you the symbol already. So this person having disease. So this person, this male, right, having a disease gene XR, right? He may, so definitely if it is male, so XY, marry with a girl, she is normal. This is normal, right? So for this, for her, what should be the phenotype? Sorry, genotype. For girl, it should be X and X and normal, right? So when he this person marry with this girl, she is normal. So what should be their offspring? So their offspring should be this. It mean two will be carrier, right? And two, like the two female will be carrier while the two male will be normal. So let's see, is it happening or no? So when we are going to do the cross between, so X R, right? This X R cross with this X, okay, and this X cross with that X, right? With everyone one time it will cross, right? And if this Y cross with X, so it will give you XY. If this Y cross with that X, so it will give you XY. So from here I know that this is male, two male, so this is XY, remember. So here we have two male, and both male is normal because there is no defective gene. You can see no small RR here. While these are two girl, and both girl having defective gene, but she, for example, this is the first girl, so she having also dominant gene, so that suppress it, and she is not homophilic, but still she is a carrier that we represent it by half shadow, right? Now let's suppose if this is F1 generation, this girl is recessive and she marry with a normal boy. So what should be the probability of their offspring? So again, you will do the same class. How you will do? You for this carrier, we have taken XR. She's female, right? So XR and X, right? It's cross with what? Cross with normal male. Cross with normal male. So XY. When you do the cross like this, so you will get these offspring. Like what you will get, one female carrier, male, another female normal, and male having hemophiliac disease. So this, by this way, you have to know about the cross, and this is just a family tree. It show you the probability. It show you what's supposed to be their upstream, and it is an easy way from which you can understand easily that who having disease, who are carrier and who are not carrier, and how the family tree are going on, right? So hope it's clear to all. Okay, if anyone having any question, they can ask. Okay, thank you so much.